Hello students, I'm Dr. Divya from TeenEinstein. So today we are going to start our class. So now we are in the second chapter, sexual reproduction in flowering plants. In the last part we have already learned about the sexual reproduction of flowering plants. So flowering plants, that means angiosperms. In angiosperms, flowers are the site of sexual reproduction. So everyone know that we know that very well about angiosperms. They are the flowering plants. They are called as flowering plants, and these flowers in angiosperms, the flowers are the site of sexual reproduction. So in sexual reproduction, male and female gamete, they'll fuse together to form a zygote. So the male gamete will form and another female gamete will form. So there is a fusion of these both gametes, and that is called as fertilization, right? So this fusion, after this fusion, it will form a zygote, isn't it? And this sexual reproduction, that includes so many events. So there are several steps under the sexual reproduction process. Okay, so we have categorized all these steps into 3 major events. So which are those major events? Mainly there are 3 major events in sexual reproduction. So which are those? First one is pre-fertilization, then second one is fertilization, and third one is post-fertilization. Okay, first pre-fertilization. Pre-fertilization means the events prior to fertilization, means before fertilization what and all arrangements we are doing or the plant is doing, that is called as pre-fertilization events. Next, fertilization. Then fertilization happens, that is the second event, fertilization, the fusion of gametes. And third event is the post-fertilization. Post-fertilization means after fertilization what and all events are taking place, that are called as post-fertilization events. Okay, so we know that sexual reproduction means it is a complex and time consuming process. We have already told it's a complex process because so many events are included in this. So it is a complex process and it takes so much of time. So it is a time consuming process also. And the duration of these process in each species, the duration of this process, the fertilization, pre-fertilization, post-fertilization and fertilization, all these events, the duration of all these events varies in each species. So compared to asexual reproduction, sexual reproduction is time consuming and complex process. We know asexual reproduction, there is no gamete formation and fusion. So in asexual reproduction, that is consuming less time compared to the sexual reproduction, right? So in sexual reproduction, gamete formation should take place, and after that fertilization. Then after fertilization, that fertilized zygote, that zygote should develop into a complete embryo. So all these things should take place in sexual reproduction. So it is a time consuming and complex process because so many events, so many processes are included under this pre-fertilization, fertilization and post-fertilization events, right? So we said in angiosperms, flowers, they are the site of sexual reproduction. So what is the meaning of that? Flower is the site of sexual reproduction. What we are meaning with that sentence? That means all these pre-fertilization, fertilization and post-fertilization, all these events are taking place in that flower, right? So flowers are the site of sexual reproduction. Within that site or within that flower, all this pre-fertilization, fertilization and post-fertilization is taking place, right? Then we discussed about the structure of a flower in the last class. And flower, it is having 4 major parts: calyx, corolla, androecium and gynoecium. Those are the four main parts of a flower. In this, calyx and corolla, they are known as non-essential words. Calyx means sepals and corolla means petals, and androecium, that is the male reproductive organ or collection of stamens. Collection of stamen, it is called as androecium, and in that stamen is the male reproductive unit, right? Stamen is the male reproductive unit and gynoecium is the female reproductive organ. Just for revising the last class, I am explaining all these things. So gynoecium is the female reproductive organ, and in this, calyx and corolla, they are together called as non-essential words. They are the non-essential words because without calyx and corolla also sexual reproduction is possible. And next two words like androecium and gynoecium, these are the essential words. These are called as essential words. Androecium and gynoecium are called as essential words because they are producing the male and female gamete, and the male and female part, without that sexual reproduction is impossible. In that, androecium is the male part and gynoecium is the female part. So without that androecium and gynoecium, sexual reproduction is impossible. Then the structure of stamen we discussed. A single stamen that consists of down the filament, stalk-like filament, and at the terminal position of that filament or at the top of that filament you can see anther, right? So coming to the anther, anther is a bilobed structure, right? Anther is a bilobed structure, and each lobe is having two theca. The bilobed anther is having two theca. If you are taking a TS or transverse section of anther, it is having four corners or four sides. So each corner is having a microsporangia. So if you are taking a TS of anther, it shows four corners or four sides, and each corner it is having a microsporangia. Okay, that means in each lobe we can see two microsporangia, right? In each lobe, two microsporangia we can see. And later these microsporangia, this will develop and form the pollen sac. That microsporangia will develop into pollen sac, right? And the structure of a microsporangia we have discussed. A microsporangia, it is almost round in shape, almost a round shape it is having, and the outer layer of microsporangia, that is the epidermis, it is single layered. Epidermis is single layered, and next layer is endothecium. Epidermis, next to epidermis is the endothecium, and then next to endothecium is the middle layers, right? And the middle layers that have 1 to 3 layer of cells. Middle layers have 1 to 3 layer of cells, and these 3 layers, they are protective layers. They have the protective function and the protection of pollen grain, and they help in the dehiscence of anther. What are all the function of these 3 layers? Epidermis, endothecium and middle layer, they are helping in the dehiscence of anther, and they are giving protection to that developing anther. And the innermost layer is tapetum. Which is the innermost layer? Tapetum is the innermost layer, and the tapetum gives, or the cells of the tapetum gives nourishment to the developing pollen grain. The cells of tapetum, that is providing nourishment, or that is having nutritive function. Okay, so in a young anther, at the center of microsporangia, a group of compactly arranged cells are present. In a very young anther, at the center you can see microsporangia. In the microsporangia, a group of compactly arranged cells at the center, right? And these cells are having homogeneous cells. All cells are of similar type. What do you mean by homogeneous? All cells are of same type. And then as the anther develops, when the anther develops, each of this will undergo meiotic division. So when anther is developing, in a developing anther, each cell will undergo. Each cell means at the center portion, each cell will undergo meiotic division, and they form microspore tetrads. They'll form microspore tetrads. So at the center of the microsporangium, you can see homogeneous tissue or homogeneous cells. All these cells will undergo meiotic division. Each and every cell of this will undergo meiotic division and it will form microspore tetrads. So each cell of this sporogenous tissue is called as a pollen mother cell, because each cell will undergo division and it will form microspore tetrads. So each cell of this sporogenous tissue, it is called as pollen mother cell, because that is developing into pollen grains, or it is called as microspore mother cell. It is called as pollen mother cell or microspore mother cell. And the formation of microspore from a pollen mother cell, it is called as microsporogenesis. So what is microsporogenesis? The formation of microspore from a pollen mother cell. From a pollen mother cell, a microspore. The formation of that microspore is, or the formation of microspore occurs from a pollen mother cell. That is called as microsporogenesis. Formation of a microspore. It is the genesis or the formation of a microspore. Then later what happens, as a part of development, these anther, it will mature and it will get dehydrated, or that will become dry. That mature anther, that means it will become dry. So that time this microspore, what is happening to that microspore? Already when it is undergoing a division, that means a meiotic division, it is in a tetrad form, means 4 cells will be there after meiosis, right? So these 4 cells will be in a joint form. So when it is getting dried, or when that anther is getting dehydrated, that tetrads, these tetrads also will get dissociated, or that will separate from each other, and it will develop into single pollen grain. Okay, so the microspore tetrad, which is formed after the division of sporogenous tissue, when this anther, it is getting dehydrated, along with that the microspore tetrads also will get separated. Okay, and it will form pollen grains. So in each microsporangium, you can see thousands of microspore, or otherwise it is called as pollen grain. So in each microsporangium, that sporogenous tissue, in this sporogenous tissue, each cell will undergo meiotic division. Means one sporogenous tissue can give rise to 4 pollen grains. Like that, inside that microsporangium, you can see thousands of microspore or thousands of pollen grains, and these pollen grains are formed inside that microsporangium. So how it will get released, all these pollen grains, how it is getting released? It will get released during anther dehiscence. So during the dehiscence of anther, this will get released. Okay, so up to this, all this we have discussed in the previous class. So now the pollen grains are formed. The pollen grains which are formed, right? So that should get released, or it should come out, right? So next we will see now that formed pollen grains, and we learn the structure of pollen grains also in this class. So we can see the structure of a pollen grain. In this, you can see in the picture that pollen grain. Here you can see the pollen grain. Here you can see this is a pollen grain, and pollen grain, that is the male gametophyte. Pollen grain is the male gametophyte. So anywhere have you seen the pollen grain? Like, see, you can see this, right? The yellow color dotted structures you can see. So if you are touching a flower, like our normal flower, like hibiscus, if you are touching that on your finger, some yellow color powder will stick, right? That is the pollen grains. That yellow color powder like structures, they are the pollen grains. And in the, you have already learned like where pollen grains, that pollen grains will be present. You can see here anther, right? And practical session also you will get pollen germination, right? So first what we should do, how we will do the pollen germination experiment. You will collect pollen grain from a common, any common plant like hibiscus and all, and you'll put a drop of sugar solution on slide, like 10% sucrose solution, something you will put, and on that you will dust this pollen grain. You will get a practical session of pollen germination. So why you are giving that sugar solution, that is providing nutrients for the germinating pollen grains. Okay, so in that sugar solution it will start germinating, and you can see small outgrowths, that pollen tube formation. You have to focus that and show to your examiner. Okay, you need to show that the germinating pollen grain. That's a simple experiment. So in practical section also you will get the pollen grain germination or pollen germination experiment. So first we should know the theory part. So now we are going to learn the theory part, the structure of the pollen grain. So generally pollen grains, you can see in the picture, the pollen grain, it is generally spherical and they are very small, very small. You can see here in the picture, and when you are taking or when you are touching that anther, it's like dust like structures, but in one touch itself you will get hundreds of anthers, hundreds of pollen grains. So structure of pollen grain we are going to learn. So it is a very small, and with the help of microscope you can see the structure, a large structure of a pollen grain. And the diameter of a pollen grain, it is around 25 to 50 micrometers. 25 to 50 micrometers. It is a very small structure. So cell wall of pollen grain, it is called as sporoderm. Cell wall of pollen grain is called as sporoderm. It's important MCQ. Sporoderm. Cell wall of that pollen grain is called as sporoderm. And the pollen grain, it is having prominent two-layered wall, right? It is having prominent wall which is two-layered. There are two layers. The outer layer is exine. The outer layer is, you can see here, the outer layer, it is called as exine, and inner layer is the intine. See, this is the inner layer, that is intine. Here outer layer is exine and inner layer is intine. So pollen grain represents the male gametophyte, and pollens are generally, they are spherical and around 20 to 25 micrometers in diameter. That is the diameter, 20 to 25 micrometers in diameter, and it has two-layered wall. That is having two-layered wall. Outer layer is called as exine and inner layer is intine. Outer layer is exine, inner layer is intine. Pollen grain structure is very simple. There are two-layered wall. Outer one is exine and inner layer, that is intine. Okay. So outer wall or outer layer of pollen grain is exine. We have already told it's exine. You can see here, this is the exine, and exine, it is made up of sporopollenin. Exine is made up of sporopollenin, and this sporopollenin is an organic material which is highly resistant. So this exine is made up of sporopollenin, which is a organic material, and this organic material is highly resistant. That organic material which is highly resistant, exine, is made up of this highly resistant organic material which is called as sporopollenin. And this sporopollenin that can withstand high temperature. Okay, sporopollenin can withstand high temperature, then strong acids and enzymes. Most of the enzymes cannot degrade sporopollenin. So exine, the outer covering, it is made up of an organic material, sporopollenin. That sporopollenin can withstand strong acids and enzymes and temperature. Almost all enzymes that cannot degrade, those enzymes cannot degrade this sporopollenin. Okay, so temperature, strong acid and enzyme, all these conditions, that sporopollenin can withstand all these conditions. Okay, because of these only that pollen grain is surviving, because the sporopollenin is having the capacity to withstand high strong enzymes, strong acids and high temperature and all. So that pollen grain can survive such conditions. Okay, so otherwise soon after releasing this pollen grain will get damaged, right? So the sporopollenin is actually protecting that pollen grain from all these temperature, acidic and alkaline condition, all this, from all these, that sporopollenin, the material of exine. Exine is made up of sporopollenin, so that sporopollenin is protecting that pollen grain. Okay, so this sporopollenin is the most resistant material. It is the most resistant material, organic material that ever known. So that is the known material, or it is a highly resistant material ever known. And that pollen grain, this exine is having prominent apertures. You can see here the exine, this outer layer exine. Exine is having that prominent apertures. That apertures are called as, they are called as germ pores. You can see here the structure, they are called as germ pores. Okay, so we have told it is having two layers. Pollen grain is having two layers. Outer layer is exine, inner layer is intine. And the outer layer exine, it is made up of sporopollenin, which is a highly resistant organic material. And this exine, it is showing prominent apertures. They are called as germ pores. Okay, in this germ pores, sporopollenin is absent. Okay, so sporopollenin is absent in these germ pores, and thus that sporopollenin, the sporopollenin is helpful in the fossilization also. Okay, because of the presence of this sporopollenin, these pollen grains are well preserved, because that acids, alkalis, enzymes and all won't work with the sporopollenin. Sporopollenin is having the capacity to resist all these things. So this pollen grain is protected very well inside this, or the pollen grain is preserved well inside the sporopollenin material, right? So these pollen grains are well preserved as a fossil, because alkali, acid, enzyme, all these things cannot degrade the sporopollenin, right? So it is so hard, so that the pollen grains are well protected from the hazardous environment, and when they are pollinated by a biotic or abiotic pollinating agent, that pollen grain, it is protected by this highly resistant sporopollenin, right? And this exine of pollen grain, it exhibits variety of patterns. You can see here, you can see variety of patterns, different patterns of pollen grain. These are the patterns of the exine, the outer layer. These are the patterns on the outer layer or exine. Okay, and that is having a lot of taxonomic significance also. And based on this, like based on the pattern of pollen grains also, studies are going on. Okay, so that's why it is having high taxonomic significance. So what and all we have learned? Pollen grain, it is having a double layered wall, the outer layer is exine and the inner layer is intine, and the outer layer it is showing prominent apertures called germ pores, and sporopollenin is not present in the germ pores. The outer layer exine is made up of highly resistant sporopollenin, but in that outer layer you can see prominent openings called germ pores, but in this germ pores sporopollenin is absent, right? And sporopollenin is absent. Just now we said sporopollenin is absent in the germ pores. And exine appears in different patterns. Different patterns you can see in exine, only in the outer wall. Okay, and that is having high taxonomic significance, because based on the pattern of this pollen grain also studies are going on. Okay, and next is the other layer, which is the second layer, intine. Intine is the second layer of the pollen grain, and the inner wall of pollen grain, that is called as intine. Inner wall, you can see here the inner wall. The outer layer we have already explained, the exine, and next is the inner wall. Inner wall is intine. And intine, it is a thin and continuous layer, but it is made up of cellulose and pectin. Intine, that is the inner layer, and it is made up of cellulose and pectin. Okay, and next to intine you can see the cytoplasm. Here you can see the cytoplasm. Just next to intine you can see the cytoplasm, and it is surrounded by a plasma membrane. You can see plasma membrane which is surrounded the cytoplasm. The plasma membrane is surrounding the cytoplasm. So what and all we have learned? Outer layer, that is exine. Exine is made up of sporopollenin, and you can see prominent apertures called germ pores, and that sporopollenin is absent in the germ pores, right? And next to exine you can see intine. Intine is the inner wall, and it is made up of cellulose and pectin. Not with sporopollenin, inner wall is made up of cellulose and pectin. And next to this intine you can see cytoplasm. That cytoplasm is covered by plasma membrane. Cytoplasm is surrounded by plasma membrane, and here inside you can see the cytoplasm. Clear? And next, inside that intine, just now we told, here you can see inside that intine you can see cytoplasm, right? And the cytoplasm of this pollen grain, it is surrounded by plasma membrane. Cytoplasm is surrounded by plasma membrane, right? Then after maturation, when that anther or the pollen grain is getting matured, a mature pollen grain, it is having two cells. You can see a mature pollen grain is having two cells. Okay, one is the vegetative cell and other one is the generative cell. Here you can see it is having a mature pollen grain. This is the structure of a mature pollen grain. It is having two cells. This is one cell and this is another cell. The bigger one is the vegetative cell. This is the bigger one, and the smaller one, that is the generative cell. So in the picture itself it's clear. The smaller one is the generative cell and the bigger one is the vegetative cell. Okay. So a mature pollen grain will be having two cells. One is the vegetative cell which is bigger, and the generative cell which is smaller. Okay. Then you can see this is the structure of a mature pollen grain. So after maturing, this is the smaller cell, that is the generative cell, and here the bigger cell, that is the vegetative cell. So in the picture you can see both cells, right? And vegetative cell is bigger compared to the generative cell. Vegetative cell is bigger, and Then you can see the nucleus of the vegetative cell. It is irregularly shaped. That vegetative cell is having a nucleus, and that nucleus don't have proper shape. It is irregular. The shape of that nucleus is irregular, and that vegetative cell is having so much of reserved food. See, you can see the cytoplasm, and compared to this generative cell, vegetative cell is bigger. So the vegetative cell is having so much of reserved food material, and it is not for any other purpose. It is for the growing pollen grain. Okay, the pollen grain is growing, so it is for the growth of that cell or the growth of the pollen grain. So next you can see the generative cell. Now vegetative cell, and next is generative cell. Generative cell is a smaller cell compared to vegetative cell, right? So the vegetative cell covers almost whole portion, and inside the cytoplasm, you can see inside the cytoplasm, this generative cell is not a separate cell. Inside the cytoplasm of the vegetative cell, this generative cell is floating, right? This whole thing, it is a vegetative cell, and in the cytoplasm of vegetative cell you can see a generative cell that is floating in the cytoplasm of vegetative cell, and this is almost spindle shaped. The generative cell is spindle shaped, and it's having a nucleus. You can see the nucleus, that red color nucleus you can see, and a dense cytoplasm. The cytoplasm of generative cell, that is also dense. Okay, so in a mature pollen grain you can see two cells, the vegetative cell and the generative cell. So compared to the generative cell, that vegetative cell is bigger and it is having dense cytoplasm, and the nucleus of that vegetative cell is irregularly shaped. Okay, and vegetative cell, it is having so much of reserve food material for the growing pollen grain. Okay, and next is the generative cell. Compared to vegetative cell, generative cell is smaller and it is having spindle shape. Okay, and that this also have a nucleus, and the generative cell is floating in the cytoplasm of the vegetative cell. This is the vegetative cell, the bigger one, and the generative cell is floating in the cytoplasm of vegetative cell. Okay, and mature pollen grain, that consists of two cells, vegetative and generative, and vegetative cell is bigger, the generative cell is smaller. Vegetative cell has abundant food reserve and a large irregularly shaped nucleus. The nucleus is large and irregular, and the vegetative cell, it is spindle shaped with dense cytoplasm and a nucleus. Over 60% of angiosperms, the pollen grains are shed in two cell stage. Almost 60% of the angiosperms, they will shed the pollen grain in the two cell stage. But in some other species or the remaining species, the generative cell, here you can see the generative cell, that will divide mitotically and it will give rise to two male gametes. Before pollen grains are shed, it will give rise to two male gametes. In that case, that pollen grain will be in three-celled stage. Okay, it will be in three-cell stage. And in majority of angiosperms, these pollen grains are shed at two-cell stage. That means while shedding that pollen grain, that will have vegetative cell and generative cell in that. And 60% of angiosperms, it is like this, and the remaining angiosperms, that means in another 40% of the flowering plants, the generative cell, that will divide, and it will divide mitotically, and this generative cell, it will divide to form two cells. That means equational division. So in the 40% of angiosperms, the generative cell divides mitotically and form two male gametes. So the pollen grain will be having one bigger vegetative cell and two small male gametes. So in total, that will be having three cells, right? So while shedding these anthers, the pollen grain will be in three-celled stage. Clear? It will be in three-cell stage in few species. Okay, and next we are going to learn about pollen allergy. So the pollen grains of many species of plants that cause pollen allergy. Okay, the pollen grain may cause pollen allergy in some people, not all people, not for all. In some people it will cause pollen allergy. A Parthenium, you can see the Parthenium plant here. The Parthenium pollen are allergic to some people. That Parthenium pollen may cause allergy in some people, and it may cause respiratory disease also, right? It that can cause respiratory diseases like asthma, bronchitis, etc. And this Parthenium, it is belonging to which family? Asteraceae family. Parthenium is belonging to Asteraceae family, and actually the Parthenium is a weed. Okay, it's a main weed in agriculture field or gardens and all, and this reduces the crop productivity. That's why Parthenium is called as a weed. It is a weed because it reduces the crop productivity. Okay, and that can cause crop production, that can reduce the crop production in a greater way, or that can absorb the nutrients compared to that crop. This weed can absorb the nutrients in a faster way, and thus in that way it reduces the crop productivity. Okay, and that can inhibit the growth of the root also. That will inhibit root growth also, and that can reduce germination and biomass growth. Everything it will reduce. This weed, that weed will grow very faster compared to the crop, because that is having the capacity to absorb the nutrients in a faster way, so that it will reduce the root growth and biomass. Then germination also it will reduce. So overall it is injurious to that crop. Okay, so the pollen grain of this plant, that can cause dermatitis. Dermatitis means skin allergy. That can cause dermatitis, so that overall this Parthenium crop, that can cause pollen allergy. So pollen grains of so many species that cause severe allergies and bronchial afflictions also, and in some people that will often lead to chronic respiratory disorders like bronchitis, asthma and all. And Parthenium, example is Parthenium or carrot grass, that cause pollen allergy. And you can see that pollen grains, they are rich in nutrients. Pollen grains are very rich in nutrients. So the pollen tablets that are made from these pollen grains, and because of they are rich in nutrients, they are making pollen tablets. Okay, so pollen tablets are made from the pollen grains, and they are rich in nutrients. And these pollen tablets are nowadays we are using as a food supplement. That pollen tablets are used as a food supplement. Same like pollen tablet, pollen syrups are also available. So the studies says that consumption of these kind of products like pollen tablet and pollen syrup, that will increase the performance of athletes. And in race horses also, say they can work in the similar way, like they can increase the performance. Okay, so pollen grains, they are rich in nutrients, and pollen tablets are used as food supplements. And a larger number of pollen products like pollen tablet and pollen syrup, such things are available in the market. And pollen consumption that can increase the performance of athlete and race horses, etc. Okay, so the pollen grains are having high economic importance. Then pollen viability and storage. So once these pollen grains are shed, that will reach on the stigmatic surface for the fertilization process, right? So fertilization means the pollen grains should reach on the stigmatic surface. And how fertilization will happen? This pollen grains will release the male gamete, right? Then that male gamete will reach inside or inside gynoecium. That is the process of fertilization, fusion of male and female gamete, right? So before losing viability, all this should happen, right? That can't survive for a long time because each pollen grain will be having a viable period. Okay, that can't stay for long time because it will lose the viability. So to bring out fertilization, that pollen grain should reach on stigmatic surface before losing its viability. So the period for which the pollen grain retain the ability to germinate after landing on stigma or stigmatic surface, it is called as pollen viability. That is having a viable period. Each pollen is having a viable period. After that, it will lose the viability and it won't germinate. It will lose the capacity for germination and for fusion. Okay, so it's highly variable and up to a limit it depends upon the climate or humidity and temperature also. Okay, the pollen viability is variable in each species and it depends upon the temperature and humidity. So in wheat, rice and all, pollen will be viable up to 30 minutes. So up to 30 minutes only, that viable period in rice, wheat and all. And after releasing, within 30 minutes it should reach on the stigmatic surface. Otherwise that will lose the viability. But in Rosaceae and Leguminosae family and Solanaceae family and all, the pollen is viable up to several months. It can stay up to several months. Right? So you know that for artificial insemination in animals and human, the storage of semen or the storage of sperm, it is done for artificial insemination, right? So in the case of plants, in the same way, it is possible to store the pollen grains of many species, because just now I said the Solanaceae family and Leguminosae family and all, that pollen is viable up to several months. So we can make use of that viability period, so that we can store the pollen grains, right? So in so many species, they are viable, that pollen grains are viable for several months. So we can utilize that property and we will store that in liquid nitrogen. So for storage, we are using liquid nitrogen. This is called as cryopreservation. So what is cryopreservation? In liquid nitrogen or in a very low temperature, we are storing this. So it is called as cryopreservation. And liquid nitrogen means it is in minus 196 degrees centigrade, right? So we can store for several years like that. So stored pollen grains that can be used as pollen banks. Okay, we can use that as pollen banks. Just like seed bank, we can utilize this property of pollen grains and we can use as pollen banks. So we can use this in crop breeding also, because the pollen can be used to produce hybrids, right? So once they are shed, that pollen grains have to land on stigma before they are losing the viability for fertilization. And the period for which pollen grain remain viable, that is highly variable. It is different in different species and depends upon the temperature and humidity. This will work. And some cereals like rice and wheat, pollen grain that will lose viability within 30 minutes after shedding, right? And in other members like Leguminosae, Solanaceae and all, that will be viable, that pollen grain will be viable up to few months. So it is possible to store this pollen grain for years in liquid nitrogen. We can make use of that property. So such stored pollen grains can be used as pollen banks, and in a similar way how seed banks we are maintaining, in similar way we can maintain the pollen banks also, and we can utilize this property in crop breeding programs. So that's all about pollen grain and pollen viability and storage. Thank you.